Thank you for the invitation. Good morning. So I'm an open vascular surgeon in Toronto, and I certainly work with a very strong interventional group there. And this morning we have had a lot of discussion about multidisciplinary treatment, and the fact that we have 3 full talks on different modalities shows that we probably don't have a very good treatment paradigm for AVM, and a lot of these patients are still left either to their natural history or suffer complication for treatment. And so one of my main research interests in basic research is the biology and the genetics of AVM. And this morning, before looking into the future, I would like to explain a little bit how we came to look at AVMs as a genetic disease caused by somatic mutation. I don't have disclosures. So we're talking about true brain AVMs, which, as you know, are part of a larger spectrum of vascular malformation of the central nervous system. And the definition we have is still a morphological definition based on imaging and angiography and macroscopic morphology, but we don't have a biological definition of the disease. So how do we approach and study the biological basis of a disease? We can first start by studying the histopathology of the disease tissue. There is certainly literature on that. We can search for germline mutation and consider the disease as having a genetic basis, and then identify which significant signaling pathway are dysregulated in the disease, for example, by analyzing the transcriptome and proteomics. And finally, we can confirm in cell culture and animal models the relevance of the identified pathways. So to summarize the knowledge so far that has been gained through these approaches is that brain AVM arise from dysregulated angiogenesis. There is a good evidence for that. We also know that dysregulation can be triggered by any angiogenic stimulus if it is combined with genetic polymorphism that cause dysregulated angiogenesis, such as in HHT, the mutation in the endoglin, ELK1, SMAD4, but also in some other susceptibility genes like short nuclear polymorphism that have been associated with AVMs. But the reality is that most brain AVM patients do not have this predisposing genetic polymorphism. Another crucial basic question in understanding how brain AVMs form is the question of whether they're a true congenital disease, whether they form during embryonic or fetal development, or they're acquired. So we know that they're, at least to my knowledge today, there is no report for in utero ultrasound diagnosing a true brain AVM. There is also a lot of women exposed to who have MRI, in utero MRIs, and there is no report of diagnosis of AVMs with in utero MRIs, but we know that we can diagnose vein of Galen malformation in utero, and this malformation is much more rare than AVMs. And there is also a growing literature of case reports reporting de novo formation of AVM in children and adults. So we can ask the question whether AVM is really a congenital disease or forms during early childhood, during still vessel maturation and growth. And so let's come back to the question, is a brain AVM a genetic disease? So we know for sure, it's firm evidence that it can be a genetic disease because it's part of the autosomal dominant diseases such as HHT, hereditary hemorrhagic telangiectasia, and much more rare genetic disease, which is also autosomal dominant capillary malformation, arteriovenous malformation associated with brain AVM, but also vascular malformation elsewhere in the body. So this is strong and firm evidence that a genetic event can cause an AVM, but we are also left with the fact that that all cells in the body have the mutation, and only a definite number of AVMs appear in those patients, and therefore there is still the question of contributed associated factor in the biology of the disease, such as environment, inflammation, hemodynamics, or a second genetic hit. So just briefly to remind you, hereditary disorders with AVM, HHT is an autosomal dominant disorder caused by germline mutation in genes of the TGF-beta SMAD pathway, which is a ubiquitous cellular pathway involved in many developmental process, tissue differentiation, but also in cancer. And there are very well-characterized mutation in endocrine, LK1, and SMAD4 that cause the disease by causing actually an overactivation of the pathway. The other genetic condition, which is caused by a germline mutation, autosomal dominant mutation in RASA1. It's a loss of function heterozygous germline mutation, which actually results in overactivation of a pathway which is called the RAS MAP kinase pathway, also a ubiquitous pathway in cellular processes involved in development, differentiation, and cancer. And basically this protein is also called P120 gap and associated with neurofibromin, which we know is mutated in neurofibromatosis type 1, and this causes overactivation, not underactivation, but overactivation of the KRAS MAP kinase pathway. Then, as I said before, are all AVM a genetic disease? Because sporadic AVM, the ones that we talk about and treat every day, are much more frequent than this rare pathological condition. And we know that there are very, very exceptional reports of familial AVMs outside of this syndromic disorder. And therefore, most of AVMs are unlikely to be caused by germline mutation. And I mentioned there are some single nuclear polymorphisms that has been associated with these susceptibility variants have not been consistently reproduced in further studies. So basically, when we started thinking about this about 7 years ago, our hypothesis was that yes, that brain AVM can be a genetic disease, but it's not a germline genetic disease. It's caused by somatic mutation, which can cause a clonal disease. And the prototype disease caused by somatic mutation is cancer, where mutation in adults, progenitor stem cell or differentiated cells can basically accumulate somatic stochastic mutation and then cause a dysregulated growth and tumorigenesis. But we also know that most of our cells, as we age, accumulate somatic mutation, but because they don't change gene function or occur in non-coding DNA, they don't cause any perturbation of tissue differentiation morphogenesis, and they just are completely innocuous. They can also lead to cell death, and then nobody cares because it kills one cell and it doesn't have any consequences. But an emerging concept in genetics is that somatic mutation can also cause tissue malformation, because the mutation in the adults, progenitor stem cell and differentiated cells will affect all the clonal progeny of these cells, and therefore affect the whole segment of the tissue or the organ which derives from the cells with the mutation, and therefore causing mutation. It sounds very logical, but actually it's a relatively new concept in genetics. If you look around you, can find many examples where this is very evident that, like superficial mutation in some cells, either pigment can alter pigmentation or cause vascular malformations, skin vascular malformation, like this port wine stain, which actually geographically map the segment of the tissue derived from the cells that have the mutation. And these examples are innocuous. And in some bad cases, when the metameric syndrome, also the same concept, where a whole developmental segment can be affected. And actually, in the last few years, there are many malformative, malformative syndromes that have been associated with somatic mutation, this very rare syndrome like the Clovis proteus, but also some CNS-specific disease, hemimegalencephaly, focal cortical dysplasia, are caused all by mutation in the PI3K, P10, AKT, mTOR pathway, also a very common cellular pathway implicated in many processes, and specific mutation in those pathways, somatic mutation, cause these diseases. There is one vascular malformation syndrome, a craniofacial venous metameric syndrome, or Sturge-Weber, has been associated in a mutation in gene NAC, which overactivates the RAS pathway that I mentioned before in this genetic AVM syndrome. So this is the emerging picture of this somatic mutation in malformative disorders. And if we think about how the vessel in the brain grow during development, we know that the brain vascularization starts around the neural tube with a vasculogenesis process that forms the perineural vascular plexus, and then from this plexus, by sprouting angiogenesis, you have these perforating vessels that penetrate the cortex in a radial way along the radial glia, and these stalk cells and tip cells then sprouts and will form these channels, and these channels, they're organized in a vertical radial way from the cortex to the ventricle, and then they will be connected horizontally, but also by sprouting angiogenesis and form this hierarchical arteriovenous and capillary network in the brain. And you can imagine that if during this process, during development, there is a mutation in one of the cells, a whole segment then that will derive from these cells will be affected and cause arteriovenous malformation. And we can speculate that if the mutation occurs very early in the development, it will be a big, malformation. And we know that they tend to be organized in a vertical radial fashion from the cortex to the ventricle, as opposed to grow horizontally across the brain in the white matter. So they have a developmental logic in their architecture. And so we can speculate a late mutation will cause a small malformation, affect less cells, and the early mutation will cause a large malformation. And in fact, there is also an emerging picture of this somatic mutation in the last few years in different vascular malformation, and we know that some lymphatic venous malformation with low flow tend to have mutation in this PI3K mTOR pathway I mentioned before, and high flow mutation like Sturge-Weber capillary malformation, congenital hemangioma, varicose venous malformation, extracranial AVM tend to have mutation in the KRAS MAP kinase pathway. So basically what we did is to look for these somatic mutation in surgical specimen of resected AVMs, compare them with the patient's blood as a surrogate for germline DNA, and look for specific mutation that occur only in AVM with whole exome sequencing. And so we have then found KRAS mutation, so mutation in the gene which is central to the KRAS pathway in most of the arteriovenous malformation of the brain. This was our New England Journal of Medicine paper last year. And so for this, we have found by sequencing and then confirmed with PCR in the majority of the patient specific mutation in G12V and G12D, so mutation in the 12th codon that are the same one that I found in many cancers, and that cause a constitutive activation of this KRAS MAP kinase pathway, where the gene, the protein is locked into an active state and has constitutive activity. And we then looked where is the mutation, where it is in endothelial cells or other vascular cells like mural cells, vascular smooth muscle, or pericytes. We have isolated endothelial cells and shown that the mutation is specific in endothelial cells of the malformation. And then we have looked at the downstream pathway, this activation of MEK, MAP kinase, genes by proteins by phosphorylation, and we have found that indeed these downstream pathways downstream of KRAS are active. We have shown that this activity actually in vitro increases the expression of gene associated with angiogenesis, has a phenotype in terms of how the cell look like and their migration, and this can be reversed by inhibiting this pathway with small molecule inhibitors. I will skip that. This has been confirmed in other studies by our collaborators in China, where they found even a higher prevalence of KRAS mutation. They found some mutation BRAF also in the same pathway. They have shown also the spinal AVMs have the same pathway, so they share a common genetic cause with brain AVM, and so we can complete then the picture of vascular malformation caused by somatic mutation by describing KRAS in brain AVMs. And this has a lot of implication in terms of translations, because small molecule inhibitors for this pathway, for the MAP kinase pathway, are present and used in clinical practice. They're called MEK inhibitors. They're used in clinical practice for some cancers that have this pathway active, such as BRAF mutated melanoma. So this opens the avenue of using some of these inhibitors to treat AVMs, and currently we're actually working on animal models in mice and zebrafish, and preliminary data, this is unpublished by some of my collaborators, show that it's possible to reproduce AVMs in mouse and zebrafish, which can be used then for preclinical studies. And actually this is already happening in humans, and this is a case report in JAMA Dermatology last year, where this group has treated a large skin arteriovenous malformation which had a biopsy. They have shown that they have a mutation in MAP2K1, so this is exactly in the same pathway, it's the downstream of KRAS, and we know that extracranial AVM tendency to have mutation downstream, and treating with MEK inhibitors, they could show quite a dramatic response of this AVM. So we're not there yet to treat brain AVM with these inhibitors, but certainly it opens an avenue to use these inhibitors, maybe also as an adjuvant therapy combined with radiosurgery or other treatments. Thank you very much.